Good morning, everybody. Welcome to another set in our Plant Phenome Journal webinar series. For today, we have Dr. Joseph Gage, who's a postdoctoral scholar at Cornell in the Buckler Lab. Very impressively, he actually has a NSF postdoctoral fellowship. Today, he'll be speaking on some past work and maybe continuing work on infield whole plant maize architecture characterized by Subcanopy Rovers and latent space phenotyping. For everybody who has questions, please feel free to type them into the chat box. I'll try and keep monitoring that. And then at the end, assuming we have time, we'll also cover questions either through chat or you can unmute and ask Joe. So without any further ado, Dr. Joseph Gage.
Great, thanks, Seth. Hello, everybody. Hope you're all having a great Monday morning so far. As Seth mentioned, I'm currently positioned in the Buckler Lab at Cornell University. Before I was here, I did my PhD with Natalia De Leon and Sean Kepler at the University of Wisconsin. That's sort of where I got into phenotyping. I built some tools there to look at maize tassel architecture, and then when I moved here to Cornell, I picked up the project that I'm going to talk about today, which involves using LiDAR data from Subcanopy Rovers to create three-dimensional reconstructions of plots of hybrid maize, and then to extract latent phenotypes from them in a sort of unsupervised manner. So I guess to start, this slide is a little bit sort of preaching to the choir in a phenomics seminar, I guess, but I still kind of like to lead off with this analogy between phenotyping and genotyping. And it's not a perfect analogy, but you know, we're sort of in this what feels like a golden age of genotyping, where sequencing is fast and cheap. It's standardized, so take almost any organism that you like, and if you can get DNA or RNA out of it, you can sequence it, and we can evaluate organisms' entire genomes in sort of one fell swoop, whether that's genotyping at hundreds, thousands, millions of markers, or whether it's all the way up to doing de novo genome assembly, which is, you know, also becoming more and more affordable. On the other hand, phenotyping tends to be expensive and slow, at least relative to genotyping. It requires people, it requires tool development, and often the phenotypes that we create and measure, whether that's by hand or in an automated high throughput kind of way, they tend to be very problem specific. So individual groups are interested in whatever questions they're interested in with whatever organism they're studying. But oftentimes, and I think this is getting a little bit better, but oftentimes specific solutions developed by a group to measure trait X and plant Y don't always translate very easily into other species, or even the same species in a different lab or with a different group who's maybe using different cameras, or, you know, where the setup is different. And finally, you know, although we can generate gigabytes, terabytes worth of image and video data for each individual, we often end up condensing it down to something more on the order of tens to thousands of data points, which maybe is for the best. I don't know what I would do with millions of phenotypic observations on a single individual, but maybe someday we'll figure out a way to handle data on that scale. And so here in the Buckler Lab, we are using autonomous Subcanopy Rovers as a way to sort of try to accelerate the amount of phenotypic data that we're gathering. And then some of the methods that I'm going to talk about today to try to make our phenotyping a little bit more generalizable. to talk a little bit about the rovers that we're using. This is an image of one on the right. They're developed by a company called EarthSense. They're based in Illinois. And this is sort of represents a kind of quickly emerging technology in the field of phenotyping. They are a nice complement to above canopy UAVs. UAVs are often flown relatively high above crop fields. Even if you have a high resolution camera on them, you can't see anything that's obscured by foliage. Whereas these rovers are in the exact opposite circumstance, where they are driving along on the ground in between the rows of your crops, and they can't see anything above them that's obscured by foliage, but they do have a really good view of the lower canopy. Working in maize, for instance, you can see things like the ears as they develop, which may not be visible to UAVs. So very nice complementary technology, I think. They are sort of similar in price point and getting cheaper as they become more popular. And they're similarly deployable. They're nice and flexible, easy to pick up and bring to a different field location, easy to pick up and walk over to another field with, whatever, you name it. The technology is maturing really quickly. The EarthSense as a company has only been around for a few years now, and they've got a sort of a fully functioning product on the market. It'll be interesting to see what other players sort of enter this space in the coming years. These robots that we're using specifically are equipped with RGB cameras. You can see those on the sides and the front, as well as a couple of LiDAR sensors. So this little gizmo with the orange cap on top is a forward-facing LiDAR, and then there's another one in the back that faces upward. And so far, the algorithm development for these rovers is also moving along really quickly. Our collaborators, Justin McGrath and Matt Siebers at the University of Illinois, have developed extremely accurate algorithms for estimating plant height from LiDAR data alone. And then EarthSense is developing in-house algorithms, or has developed in-house algorithms for stand count, stem width, leaf area index, and has some others under development right now. So we're excited to be working with these rovers. But they're still a really good example of kind of what I see as a blessing and a curse in phenotyping right now, which is that affordable equipment is very much becoming the norm, or already is the norm, whether that be cameras, video, various different flavors of sensors, and the computing power to store and process all of that data. It's becoming super, super cheap, which is fantastic. But at the same time, it means that we've sort of got this massive influx of image and sensor data, which seems to be more than I think we can effectively make use of right now. So as I mentioned before, frequently individual labs are interested in their own particular crops and their own particular questions within those crops. And so maybe you gather terabytes or petabytes of image and sensor data in order to try and answer a particular question. And whatever aspect of that data is useful for answering that question gets utilized. You could think of this in terms of, like, say, hyperspectral imaging. Maybe particular wavelengths are of interest for predicting a certain phenotype, but the rest of those wavelengths are go unused. And maybe that is because they are actually useless, but maybe there's interesting data in there that could be used. And so the question that we've kind of started thinking about is really how can we set up the way that we're doing phenotyping in order to maximize the utility of all of this data and really kind of wring as much information out of it as possible in an efficient manner. So the way I sort of see the status quo right now is that we are able to generate massive amounts of data, and then we have to figure out a way to convert that into our trait of interest. And very frequently those traits are the types of things that we've been measuring for decades already. Not always, I'm generalizing a little bit here. And for good reason. So things like plant height or flowering time or various other traits that have been designated as important by, say, plant breeders over the years are the norm because they're informative. So not to diminish those in any way. But I think that in addition to finding ways to evaluate these phenotypes in novel and more high throughput manner, we could also be thinking about ways to generate traits that maybe we haven't thought of before. And so in our group, we are thinking about ways to replace the people translating data into traits with computers, and to leverage machine learning to identify aspects of our data that are most informative, say about the difference between individuals in a field or the difference between genotypes in a trial. Obviously, this isn't something that should just be done all willy-nilly. We need to sort of think through how we are using machine learning to create new traits, and we need to try to figure out whether they're actually telling us anything useful, which is kind of going to be the focus of the project that I am talking about today. So if we do this in an informed manner, maybe we're able to gain insight into how plants work and apply that to breed them better or to learn things about biology. But in order for these sort of machine learning identified traits to be useful to us, they need to be automated or semi-automated, such that we can produce them quickly and easily without too much interference, or without imposing too many of our own ideas of what's important and what isn't. They need to be generalizable. So this is kind of getting back to what I was talking about earlier, how individual groups are asking their own individual questions. But what if we had sort of a universal phenotyping platform where you can gain insight about whatever it is that you're working on without having to develop tools specific to your questions. And then finally, they still need to be applicable, right? So we need to be able to translate whatever's coming out of these models into actual biological insight, or into actual breeding decisions. And so these 3 points may sort of feel like they're a little bit at odds with each other in some ways, but I think that at some point we may be able to get there. And so the project that I'm going to talk about today, sort of a first stab at working in that direction. And I don't think I have hit the nail on the head perfectly, but you know, these are the kinds of things that we're thinking about as we develop our phenotyping pipelines here. So the sort of case study to explore this question that I want to talk about today is to answer this question up at top. So can we use machine learning to describe plant size and shape in Maize. I say size and shape because we're using LiDAR data, which gives us information about the sort of physical space occupied by the plants in the plots that we're driving through. I'm only going to talk about LiDAR data today. We did not in this study make use of the camera data on the rovers. That is a completely separate stream for now. So we use these Earth Sense rovers. We drive them in between the rows of a sort of standard hybrid trial field. And the LiDAR allows us to make three-dimensional reconstructions of every single plot in the field, which is just mind-blowing to me. I mean, I think this is really, really cool. It takes very little pre-processing to go from raw data to a point cloud like the one that I'm showing you on the upper right-hand corner here. But then we're sort of stuck with this question of how to analyze this data. And I think it's sort of a perfect case point for the arguments that I just made for using machine learning and phenotyping, because it is inconsistent and it's noisy. So we get varying quantity of data back from each plot, just sort of depending on how many surfaces the LiDAR beams hit as it's driving through and how fast the rover is moving, how much it gets jostled around, things like that. Then the data is inherently very noisy, and it can be hard to figure out exactly how to smooth that noise out or remove it. But at the same time, this data stream is really information rich. I mean, like I said, we have a three-dimensional representation of every single plot in the field. So in theory, anything you could want to know about plant architecture is contained in this data set if you can find a way to pull it out. And that's sort of the crux, right? So rather than try to pull out the kinds of traits that we've measured traditionally in the past, we attempt to use machine learning to allow a computer to decide what aspects of that point cloud are most interesting or most differentiate genotypes from each other. I should point out before I go on, for those who are interested in the germplasm that we use for this study, this was one location in a larger multi-institution collaborative project called Genomes to Fields. And so we surveyed about 1,600 plots of hybrid maize that were derived from 3 biparental populations crossed to a couple different testers. And so in that regard, it does sort of resemble the kind of field setup and experimental design that you might expect from a breeding yield trial. So, as I mentioned before, we've got these three-dimensional LiDAR point clouds. And the first question is sort of how to deal with the amount of noise and the inconsistent quantity of data that I already described. It's really difficult to segment individual plants out of these point clouds, or at least I think it is. Maybe if there's somebody out there who thinks they could do a good job of that, get in touch with me afterwards, because I'd love to hear from you and love to work with you. But it's kind of hard to tell which points belong to which plants. And so what we do, instead of working with the data in 3D, is we summarize it into 2 dimensions. So if you consider the 3 dimensions of our plot here to be lateral, vertical, and then the length of the plot, we summarize by collapsing this along the plot length and taking the density in a number of different bins from top to bottom and left to right. Then we fold this in half on itself across the center of the plot. And the result is that we get a density map shown here on the right that represents essentially what we're thinking of as an average plant in the plot. And so obviously it's no longer crisp and well-defined anymore because it's been averaged over both rows of the entire plot. But nonetheless, you can still see sort of where the stalk is, where some of the leaves are. Up top, you've got more leaves or maybe a tassel, kind of hard to tell in this example. And then these density maps are treated as images, and these are sort of the base unit that we use to build our machine learning models and to extract latent phenotypes. So in order to try and train models that can give us meaningful underlying phenotypes without too much interference on our end, we use two different methods, principal component analysis and an autoencoder, which I'm going to talk about in a minute, to create what we refer to in the manuscript as latent space phenotypes. This is a term that we borrowed from a paper by Jordan Ubens and Ian Stavnis. Sort of came to find out that we're using the terminology in a slightly different way than they are. But nonetheless, the sort of underlying point is that these are phenotypes that exist in sort of a more restricted space than the original density maps, and that can be treated as normal phenotypes. For those who are familiar with Dan Chitwood and Christopher Topp at the Danforth Center, they wrote a paper together in 2015 who referred to the same sort of idea of underlying latent descriptors of phenotypes as cryptotypes. And this idea of pulling latent descriptors of an image of something out also goes back all the way to the early 90s with eigenfaces. And so what we're doing here is certainly not novel. Lots of other people have thought about it. But what we're doing is kind of trying to take these ideas, apply them to a real field setting, and evaluate whether these latent phenotypes that we create or that a computer creates are worth anything, evaluate them for their potential utility. So before I talk about evaluating those for their utility, I'm going to give you a brief overview on autoencoders. I'm going to assume that most of the audience is familiar with principal component analysis, but autoencoders I found less people to be familiar with. And also the way that we built our autoencoders was slightly different than the way they have been traditionally built. So I'm going to talk about that a little bit before going into the results. So an autoencoder is a type of neural network where the goal is to compress, in this case, an input image down to a reduced representation in a latent space in such a way that that latent representation still contains enough information to reconstruct the image as well as possible. So you can think of this as sort of a flavor of image compression. But the idea is so say your input image is a stalk of corn. It goes through a series of layers shown by this gray box on the left-hand side, which the goal of which is to compress it to a vector or a matrix of latent space, in our case, latent space phenotypes. So these values in the dark gray box are what we use as our phenotypes. But they're created in such a way that they can then sort of be, the process can be reversed and you can reconstruct the original image. Depending on how good the video is here and how well this is coming through, you may or may not be able to see that the reconstructed image is a little bit blurrier than the original, and that the contrast is a little different. So this is not a lossless process. We do lose some information, but if the network is trained correctly, you are able to reconstitute the most relevant features of the input image, or the most important features, as well as you can. So that's sort of your standard autoencoder architecture. We did things a little differently because in the example that I just showed you, the autoencoder learns to represent the original image, right? So that the output, this corn plant, is as similar to the original corn plant as possible, given that it's being compressed. But in our case, we don't want to reconstruct our input image perfectly. There are elements that we want to ignore. So for example, if you drive a robot through the same plot a bunch of times, you're not going to get the same exact density map out every time that you do. And this is just because the wind is blowing, the leaves are moving, the LiDAR is sampling different points within the plot. And so instead, you'll get two things that look similar in some regards, but differ in other regards. So on the left-hand side here, we have these two images that I've noted as rep 1 and rep 2. These are an actual example of technical replicates from driving through the same plot twice. And so we want this model to learn aspects of the image that are inherent to that plot without learning the noisy bits, or the bits that we don't care about. And so what we do is we train the model in such a way that it compresses these technical replicates separately into a latent space, then averages those latent representations together and uses that average to recreate a reconstructed image. And what you can see is that after the network is trained, it creates an even fuzzier version of these input images that sort of bears a passing resemblance to both of them. And what we're hoping is that in doing it this way, we're teaching the model to learn the aspects of these technical replicates that are important while ignoring things like, like leaf motion or the position of the rover within the plot as it drives through. So we use our autoencoder and we use principal component analysis separately to derive latent space phenotypes. But that's really only the beginning. Once we have these latent phenotypes that are somehow chosen by computer models, the question is whether they're any good or whether they're at all useful. And so in order to evaluate them, we checked their heritability, which should give some indication of whether they could be useful for selecting on in a breeding program, or for, say, taking replicated measurements and doing genetic mapping. And then we also used them to build models that predict manually measured phenotypes and see how good of a job they do predicting those sort of ground truth, traditional type phenotypes. So heritability first. what we have here is a plot showing the heritability of some manually measured traits. These are the sort of standard types of traits that are measured in corn. So we have things like plant height, time to flowering, leaf counts, some harvest phenotypes like test weight and plot weight, and some lodging phenotypes. And you can see that these run the range in broad sense heritability from about 0.75, 0.8, all the way down to about 0.25. This is just to give you sort of a baseline to compare the latent phenotypes to. The latent phenotypes we find range from not heritable at all up to heritabilities of about 0.4. And while that is certainly not through the roof, those heritabilities in the 0.3 to 0.4 range are fairly comparable with sort of middle of the road heritability traditional phenotypes. So we think that this is really quite promising and quite competitive with standard measures of phenotyping, especially given that traits like plant height and flowering are traditionally very heritable, and they're sort of, I don't know, you could almost describe them as single dimensional in that, not in that they literally are because they're a single value, but if you think about sort of the whole space of plant phenotypes, plant height and flowering are really just sort of one strong aspect of that space. Whereas these latent phenotypes that we are generating by PCA and with the autoencoders are really describing plant biomass as a whole within the plot and within our single averaged plant that we've created in the density maps. So we're pretty happy with these heritabilities as a starting point. The second metric that we use to evaluate their quality or utility was how well they can be used to predict manually measured phenotypes. So you could imagine that we may have made latent phenotypes that are heritable, but still somehow are picking up on some sort of weird signal in the density maps that's not actually related to plant size and shape. I can't imagine exactly what it would be, but we wanted to make sure that there's still information about plant architecture contained in these latent phenotypes. And so we use the same set of sort of standard manual phenotypes that I just showed you before in the heritability plot, and built PLS models to predict them from the latent space phenotypes. And what we see is that these latent phenotypes can do a really good job of predicting traditional manually measured phenotypes, ranging in prediction accuracy from about 0.75, 0.8 down to around 0.25. In this plot, the accuracy on the y-axis is just measured as the correlation between the predicted and the measured phenotypic values. So that's fantastic. That's also really good news. That gives us some indication that there's information about plant architecture contained in these latent phenotypes. But one thing kind of gave us a little bit of pause. I was giving a talk on this maybe a year ago or before we submitted the manuscript for it, and somebody asked me how phenotypes, latent phenotypes created from LiDAR data, which really only describes the physical location of plant surfaces within the plot, can predict completely non-architectural traits, things like flowering and harvest traits like grain moisture, plot weight, test weight. That's a really good question. I wasn't sure at the time, but we hypothesized that these traits are similarly predictable using the other more architectural traits, things like plant height, ear height, and then also sort of agronomic traits like lodging and stand count and leaf counts. So if you consider the traits in the red boxes as being non-architectural, and for the sake of simplicity, we'll refer to all the other traits as being architectural traits. What we did is we just used the exact same flavor of PLS models to predict these non-architectural traits using the architectural traits. So that's shown in green now. And we see sort of similar prediction accuracies between the latent, regardless of whether you use these latent phenotypes to predict non-architectural traits or other architectural traits to predict them. So there's no sort of black magic going on that we're able to predict things like flowering time from LiDAR data. It's just that there's sort of shared signal between these physical plant architecture traits and things like flowering, grain moisture, et cetera, et cetera. So after all of that, something that I've deliberately not addressed yet is the fact that through all of the previous figures that I showed you, both PCA shown by yellow and the autoencoder shown in purple give really, really similar results. We are fairly confident that both of these are just serving to pull out the greatest source of variation between the plots. And although the traits, the latent traits tend to be slightly different between the two methods, they seem to be capturing sort of the same quantity of information. Given all of that, PCA is definitely simpler to run. It's a little more intuitive than the autoencoder. And so we sort of consider that our standard to beat. It was included because it's straightforward and tends to give good results frequently. But we don't think that this is cause to abandon this autoencoder idea entirely. There may be other neural net model architectures that are able to beat PCA. And if people have ideas about how to structure our networks in a way to sort of wring more information out of them, I'd be really happy to hear about that. Again, this study is really just a leaping off point. And it's worth discussing a little bit some of the shortcomings of these latent phenotypes. For instance, these models were built entirely with a single set of germplasm in one field season. We have no idea yet if they will hold up with data collected in subsequent field seasons or with different populations. My prediction would be that we will still get some signal, but that it won't be as strong. So the heritabilities will be lower and the prediction accuracies will be lower. So some really strong thought about how to create models that generalize across populations and across field seasons would be helpful and is going to be important moving forward. They were trained at a single time point, and so I don't anticipate that they would work very well with longitudinal data collection. If you put a LiDAR scan of some seedlings through these models, I don't think you would get anything particularly interpretable out or useful. So do we just train different models for every point in a plant's development, or do we think of a smart way to train things that are sort of temporally aware? That's another thing that we're thinking about moving forward. And finally, I mentioned this briefly a slide or two ago, but this was pointed out to us by a reviewer. These methods, both PCA and the autoencoder, are really just identifying the largest sources of variation between our density images, which is sort of the point. But if there are sources of variation between those density images that are due to the way the data was collected or due to systematic error in particular plots, the orientation of the rover, things like that, we need to be really cautious that we're being aware of that possibility and that we're compensating for it. Whether that's after the fact in data processing or before the fact in data collection. And finally, from a sort of a practical standpoint, we don't have a great feeling yet for how to interpret these latent phenotypes, much less how to apply them. So I would sort of argue that you need to have some understanding of a phenotype before you start selecting on it. In the case of PCA, the principal component analysis derived latent phenotypes, we can get an idea of what they represent. So we can sort of project the eigenvectors back into the shape of the image and sort of get an idea of whether each principal component is highlighting the stalk or the leaves or the top or the bottom of the plant. But when we do that, it's not as clear cut as it could be, which is good on one hand, because it means we're sort of pulling variation from all over the plant. But on the other hand, it's hard to interpret. These latent phenotypes are heritable, so you could select on them in a breeding program if you knew which ones corresponded to the types of plants that you wanted to select for. But I don't think we're anywhere near the point just yet where they'll become a standard part of a breeding program. But you can imagine in the future that UAVs will fly over every field and UGVs will drive through the rows in every field, and we will be able to make breeding decisions based on those. But just to reiterate, that was a very doom and gloom slide, but there is still value in them. And for that, I just point back to these two metrics that we use to evaluate them. They're heritable, and they do contain information about phenotypes that we measure traditionally. So there's some value here, and I think that our work is really cut out for us moving forward to keep kind of pushing the envelope in this direction. This is all stuff that I've sort of already said, and we're pushing up against 45 minutes, so I won't talk about that much. This as well, again, we don't know how these models fare on new data, but we're working on it. As far as future work with just rover-based phenotyping in general goes, regardless of whether we analyze the data with this latent phenotyping approach or just sort of measure traditional phenotypes from the data we collect, these rovers have been really promising, and they're offering really kind of unprecedented temporal resolution for this scale of field experiment. UAVs offer similar temporal resolution, but the rovers are collecting completely different types of data. And so just thinking about phenotyping as we move into the future, these types of technologies are allowing longitudinal phenotyping to become the new norm. So thinking about ways to really incorporate temporal aspects into our phenotyping and our data analysis, I think is going to be really important. We're sitting on a whole lot of data collected by these rovers in 2019. All of the work that I presented was based on data from 2018, but I wasn't able to get it processed up early enough to talk about it today. But we have it, and we're working on it. In addition, these rovers offer unprecedented spatial resolution. So there was a time when you could only had the time to measure a single plant in each plot when phenotyping, but that's really not the case anymore. This image on the right-hand side is just a still taken from a YouTube video produced by EarthSense, where you can see that they're measuring the stem width of every single plant in a plot as they drive by it. And not only are they measuring every single plant, they're measuring every single plant multiple times. So I think we are being offered the ability to have much more precise and accurate phenotypes. But there's also going to be opportunity to ask lots of really neat, new, interesting questions about things like phenotyping single plants, or how variability within plots or even within plants affects their growth and their interactions with each other. So it's an exciting time to be working in phenotyping, and I think there's a lot of really neat work yet to be done. Again, this is stuff that I've already talked about, but these latent phenotypes certainly don't stand by themselves. And so we are trying to think of how to combine latent phenotypes and rover-based phenotypic data with other streams, whether they be from UAVs above the canopy, genomic information, whether that's SNP markers or expression, and then also things like weather and agronomic information. And the Genomes to Fields initiative that I mentioned earlier is, I think, going to be a really nice platform to do some of this work, combining these types of data streams. So with that, I've talked long enough. I've used up almost my whole hour. I'd like to thank all the members of the Buckler Lab as well as the Gore Lab here at Cornell. This has been a joint effort between the two of them, as well as the numerous people who have helped drive the rovers in the field and helped with data collection in that way. I thank EarthSense as well. They built the rovers that we're using, but have been extremely helpful in troubleshooting and taking our feedback into account in successive iterations of the rovers. So it's been really, really a pleasure working with them. And of course, our funding sources. I particularly want to point out that a lot of this work was funded by ARPA-E and the US DOE. And that my funding, although it currently comes from NSF, was largely provided by USDA when I was working on this project. So with that, I've certainly talked enough. I would love to take any questions, comments, suggestions from the audience. So thank you.
Okay, well, thank you, Dr. Gage. We'd like to open it up for the next 12 or so minutes for questions, which you can write in the chat section. And I actually haven't been checking the chat section like I should have, but I don't see we have any chats there. And you can make it directed at either Joe, myself, or everybody, and we can read. Or you can unmute and turn down your volume and ask a question after introducing yourself. So while you're getting your questions ready, I'll just make a brief plug for the Plant Phenome Journal that lets us host these webinars. And we are an open access society journal dedicated to advancing issues and you know, I think this is the first article in the journal that really has mentioned latent space phenotyping, which is clearly going to be a hot and important growth area as we move forward. And so we're still trying to push the frontiers of phenomics, just as Phenome 20 meeting just did last week in Tucson, and as the North American Plant Phenotyping Network is doing. And so I think Joe's doing a great job of showing us what the future is. Joe, I got a number of questions here myself. I'm going to kick off the first one here, and while people are getting the rest of their questions ready. When you were showing your principal components and your autoencoder, I noticed that principal component 2 actually explained quite a bit more information than principal component 1, which to me is somewhat counterintuitive. Any explanation for that?
Yeah, so you're talking about here?
Yes, sir.
Yeah, so although principal component analysis works in such a way that PC1 explains the greatest variance within the input data, that doesn't necessarily mean that it is the most heritable. So we think that in cases like this, PC1 is probably picking up a lot of variability between the images, but a lot of that may be due to things like the position of the rover relative to the plants, or just the density of points within the point cloud, things that are contributing to differences between the images, but that are not necessarily related to differences between genotypes in the field.
Okay, that makes sense. You still got to be a little surprised that principal component 16 was the third highest, though, right?
Yeah, a little bit. And if you sort of pick these apart and look at which parts of the genome, or which parts of the genome, which parts of the density maps they're highlighting, you see that PC16, for instance, I think is much more fragmented than PC1 and PC2. So it may be picking up signal related to things like leaf position, or something kind of a little more nuanced than what we see in the earlier principal components. As I also mentioned, this is data from a single field season, so it's entirely possible that that linear combination of pixel values is not it's not worth much when you add in new data. So we'll see.
Okay, well, thank you, Joe. We've got a question from Dr. Dan Northrup. Is it a conflict that you can predict a highly heritable trait like height with latent space phenotypes that has lower heritability?
Yeah, that's a good question. In this case, no. So it's important to point out that we're not predicting the true genetic value of these genotypes with the latent phenotypes. We are only predicting the measured values. So in that case, it's not surprising to see prediction accuracies like these that are higher than the heritabilities, for instance. What would be interesting would be to see how well we can predict the BLUPs for these genotypes rather than the raw values.
Okay. That's the only question I've got in the chat. I don't know if you see any in your chat, Dr. Gage.
Oh, let's see. I am a little bit Zoom illiterate, but no, I don't see anything.
Okay. Would anybody like to unmute and ask a question?
If there aren't any questions now, I'm also happy to chat over email, or I'll be at the Maize genetics meeting in a couple of weeks. So happy to talk with people in person.
Great. Well, I've got two more.
Okay.
One is, you were talking about how, for instance, a structure and point cloud might somewhat, share information. And I just wonder if you'd taken a look at Rinson's paper on phenomic selection and looked at some of those methodologies or the philosophy behind that.
I have not read the paper, but I've heard the term. So I'm not gonna wade into that water in case I say something wrong. But I would love to read it.
Yeah. Because I think that's probably some of what's going on here is your ability to predict other genotypes is probably due to, just like genetic markers show relatedness, so can some of the phenotypic traits, right?
Sure. Yeah, absolutely.
I'm seeing a number of people are joining. I guess I didn't do a very good job of getting the time zone right this time. So.
No.
Last question I got for you. I think when you were showing accuracy, like for instance in the slide you got up right now, that's some form of cross-validation. How did you do the cross-validation?
Cross-validation was done tenfold based on genotype. So drop out all the observations that belong to 10% of the genotypes, build the model on the other 90% and predict that 10%.
Okay, and you dropped out both reps of the genotypes.
Yeah, exactly. Very careful to do it on a genotype basis rather than an observation basis.
But it was done randomly across all 1,600 plots and not within some subset of.
Correct. Yeah, no, we didn't subset within biparental, for instance, or anything like that.
Okay.
Yep.
Oh, Justin is saying I gave the time as 12 p.m. CST. The one email that I accidentally sent out that said that had 12 CST and 11 CST in the same email. So my apologies. Sorry, Justin. Try not to bombard everybody with too many emails.
Yeah, if you are just joining and missed the talk, I'm happy to give you the condensed version at another time.
Well, it's also been recorded, and hopefully I'll get it up on the YouTube site in the next 24 hours would be my hope. We'll see.
Okay. Great. Any other
questions?
Comments, questions before I split?
Okay, well, thank you, Dr. Gage. Great job. Really interesting work, and keep it up. Look forward to seeing what's coming out next.
Thank you, Seth. It's been a real pleasure, and thanks everybody for tuning in.
Thank you, guys. Bye.